Welcome to my channel Adhyan with Flow. Let's start with structure of contractile proteins. Now, what is contractile protein? Guys, they are main component of thin and thick filament. Which kind of microfiber responsible for forming thin filament? The name goes to actin. Which myofibril form thick filament? The name goes to myosin. Myosin form thick filament and actin form thin filament. We all know this. Now let's start with some brief introduction about actin. This is made up of filamentous actin. Filamentous actin. They are, like their name, they are filament in structure. They are fibrous in structure. They coiled together. And each F actin is polymer of G actins. There are two types of actin. I am writing here, there are two types of actin. The first type is, this actin made up of two types of component, F component and G component. F component, we can call it as a filamentous part of actin. And G component, we can call it as a globular part of actin. Clear, guys? Now, to filament of another protein, tropomyosin, also run close to F actin throughout its length. We want to say that this is the actin filament, this is the F filament, this is the F filament of actin. Okay, now the G protein like tropomyosin covered the filamentous part throughout the length. Now I'm writing here, this is the filamentous part of actin and this is the globular part of actin. Clear, guys? Clear? Very clear. Both are the part of actin. This is arranged in this manner. Look at here. They are coiled. They are coiled together. They are coiled together. They make a helical structure. This is the one actin filament. This is the second actin filament. This is the third actin filament. And these are tropomyosin. A complex protein, troponin, is distributed at regular interval on the tropomyosin. He want to say that this is the actin. Okay, now I'm drawing this thing, tropomyosin, like in this way. Alright, guys, if I zoom this thing, they look like in this shape. And troponin located on tropomyosin. Troponin located on tropomyosin. Is it clear, guys? Is it clear? This is the actin part. This is the tropomyosin part. And this is the troponin. Alright, guys. This position found when cell is in resting phase. Resting phase. There are no stimulus. Tropomyosin and troponin occupy all the space of actin. Now let's talk about some brief introduction about myosin. We complete our discussion about actin. Now switch towards myosin. Myosin form thick filament, we know. They are also a polymerized protein. Many monomeric protein called meromyosins constitute one thick filament, and each merosin has two important parts of a globular head and short arm. Okay, I'll teach you here. Look at this diagram. This is the myosin structure and this is the actin's structure. Alright, guys, these are troponin and this is tropomyosin and this is fibrous part. Alright, guys. Alright, now let's discuss about myosin. Myosin made up of monomer. Which kind of monomer? They are made up with meromyosin. And what is the meromyosin? They are double helical structure. They contain head part and tail part. We can call it as a heavy meromycin and light meromycin. They also have binding sites for actin. Look at here. This is the binding sites of actin, and this is the binding sites of ATP. They are the binding sites of ATP. This is the head part, and this is the tail part. The tail part is light, and head part is heavy. Alright, guys. Now, this is all about myosin. This is all about myosin. This is the heavy meromycin region. This is the heavy meromycin region. Alright, and the lighter meromycin region. Heavy meromycin component, the head and short arm project outwards at regular distance and angle from each from the surface of polymerized myosin filament is known as fossa. This whole portion is heavy meromycin. They have two outgrowth. Okay, the globular head is the active ATPase enzyme and has binding site for ATP and active site for actin. These site, actin binding site attach with actin and ATP binding site attach with ATP. This is all about actin and myosin. Here's mechanism of muscle contraction. How muscle will contract and relax. There is nothing. This is the sliding movement of thin filament over the thick filament. This is the important line. Mechanism of muscle contraction. What is the muscle contraction? This is the sliding of thin filament over the thick filament. This is the thick filament. If contraction will there, then thin filament cover the thick filament. Thin filament cover the thick filament. This is the actin. This is the myosin. This is the actin, and this is the myosin. If cell is relaxed, no stimulus, then this orientation like this. Clear, guys? If muscle contract, they also contract. And here myosin's head. Clear, guys? Here is head of myosin. They support for sliding. When muscle contract, they attach with actin. And when muscle is relaxed, these are head part, guys. These are head part. This is the head part. They are like this. Alright, guys. This is the myosin part, and these are the head part. They never attach in relaxed movement. And when contraction is occur, they attach with actin. They are actin binding site here. They are actin binding site attached with actin. Alright, guys. Look at here. This is the explanation. This is the whole explanation about how muscle contraction occur. This is the myosin filament, and this is the head part of whatever I explain here. This is written here and here also. Okay. Myosin. This is the head part of myosin. Myosin attach with actin by their actin binding sites. I'm writing here. This is the step 1. I'm writing here by the flow chart. First, myosin had attached with actin by their actin binding sites. Clear? Myosin have actin binding sites and they attach with actin. The first step is clear. Now, what is this? ADP and PI. Now, second step is first step and second step. Now, what happened next? When myosin had attached to actin, then attachment activate ATPase enzyme. So there are use of ATP, and this ATP break down into ADP and PI. If we want to break this ATP, then ATP convert into ADP plus free phosphate molecule, and if we want to break down ADP, then ATP change into AMP and free phosphate. What is ATP? Adenosine triphosphate. What is ADP? Adenosine diphosphate. What is AMP? Adenosine monophosphate. This is the energetic form, energy form. This is less energy form, and this is lowest energy form. They give energy. We require energy for muscle movement. Clear, guys? We require energy. Then what has happened? Here is actin. Here is actin. All right, and this is the myosin. This head attach with actin. Head part of myosin attach with actin. And now what happened? There is activation of enzyme. There is activation of enzyme. There is activation of enzyme. What kind of enzyme? ATPase. ATPase. So here ATP is required. Now let's move further. Step B. Now myosin had cross bridge with actin. Myosin had cross bridge with actin. They now finally attach. Now ADP also attach here. Now at C position, this cross bridge push their self through center. Here, forward direction. They push myosin head and complete actin towards the center. This direction. This direction. This direction. They push their self to this direction. Means cross bridge rotate forward. Okay, we can also call this is a technique of power stroke. Myosin had pushed their cell forward because of cross bridge. Now this ADP molecule is released and PI molecule is released. Now ADP plus PI combined and they form ATP. Now when ATP come, myosin and actin binding detached. Myosin and actin binding detached after entry of ATP. Now this actin move forward. Now this head part is removed and attached with another like this, and this forward. This, look at here, this is cross pitch and forward. So first position is this, second position is this, now third position is this, and here long actin filament, and this is the fourth position. First they bind myosin head with this position, ATP come, they remove. Then bind to second position, ATP come and they remove. Then bind at third position, ATP come and they remove. Then bind with fourth position, ATP come and remove. So they push actin towards the center, towards the forward. So this is the whole mechanism of contraction of muscle. And this process we require Mg plus 2 as well as calcium plus 2. Without these iron, the muscles can't move. Okay, the calcium is necessary for muscle contraction. Calcium is necessary for contraction of muscles. Whatever I explained here, this is written here and here and here also. Okay, look at here. This is the relaxed form of sarcomere when no stimulus. And this is the Z line, relaxed form. This is the Z line, relaxed form. This is the A band zone, and this is the H zone. This is the dark A band zone. This is the I zone. This is the light, dark. Now, if stimulus is come, then muscle start contracting. So this actin filament move forward. Why? How? Because myosin head push forward. Myosin head push forward in this direction. This is location. Look at here. The previous location of actin is this, and now they move forward. And now the previous location of actin is this, and now they move towards the center. This is the maximum contracted stage, and this is the contracting stage, and this is the relaxed stage. So here no H zone, the diameter of sarcomere is decreased. Diameter of sarcomere is decreased. All right, the diameter of I band is also decreased. The diameter of A band is increased. Look at here, this is a whole dark zone now. Whatever I explain, this is written here. All right, guys. Okay, next important word, myoglobin. We all know that what is hemoglobin. They have hemoglobin. Where hemoglobin is present? In RBC. Where myoglobin is present? In muscle cells. They use ATP. They also use ATP. They bind with one oxygen. They bind with 4 oxygen. Means they transport oxygen to muscles, and they transport oxygen in complete body. So that's all for now, guys. we will meet in our next video. So subscribe my channel. If you like my video, then please subscribe and support me, guys. Thank you for listening me, for supporting me. Thank you so much.